Okay, beta, in previous class we studied about how do the enzymes work, right? And today I'm going to revise this and then we will study about the effects of temperature and pH on enzyme, activity of enzyme. Here you can see this green one is the enzyme, right? And I told you it has an active site. This depression is actually showing the active site, and this purple or pink here is the substrate. You can see that the shape of the active site is complementary to the shape of the substrate, and the substrate will fit into this active site, and in next step enzyme substrate complex will be formed. This is the enzyme substrate complex in which the active site is occupied by the substrate. Then in third step, this substrate will be converted into product by the enzyme. Reaction will occur and the substrate is converted into product by the enzyme, and in fourth step products are released. These two pieces are showing the product, and enzyme is also released as it is, which can be reused or used again in the next step. This is the reason that enzymes are required in very small amount. Understood? Okay, beta. Next is, this is the first topic of our today's class, and this is about the factors affecting the enzymes or rate of enzymes. Actually, every enzyme works on an optimum temperature, or enzyme's activity is maximum on a temperature. That temperature is known as the optimum temperature, and this temperature is different for every enzyme. All enzymes will not be working at just one temperature. They will be working at different temperatures. Those enzymes which are working inside the human body, they work at 37 degree centigrade. So 37 is the optimum temperature for the enzymes of the human body. In the same way, enzymes which are working in plants, they would have different optimum temperature. Optimum temperature is that temperature at which the activity of enzyme is maximum. They are showing that maximum activity or maximum products are actually being produced. That is the one temperature, and that's known as the optimum temperature. And every enzyme has its own optimum temperature. All will not show the maximum activity at one temperature. All enzymes will have different temperatures. Now we are going to see how actually this temperature affects the activity of enzyme. At low temperature, here you can see at 0 or at low temperature, enzyme is inactive. Inactive means that it is not active, or it is not actually converting the reactants into the products. But if we are going to increase the temperature, it will become active, right? As here you can see in the graph, we are increasing the temperature. Here you can see we are increasing the temperature, and this upward graph shows that the activity of the enzyme is also being increased. On y-axis, you can see there is the rate of reaction or activity of enzyme, and on x-axis there is the temperature. As we are increasing the temperature, activity is also going upward, or it is also increasing. So it shows at 0. Here you can see at 0 temperature, the activity is also 0. As we are increasing the temperature, the activity is also going upward. And at this particular temperature, which is the optimum temperature, the activity of enzyme is maximum, right? After this temperature, if we further increase the temperature, you can see from 3.7, we have further increased the temperature to 5.5, and its activity has it has gone down. So this shows that when the temperature is increased beyond the optimum temperature, the activity decreases. This downward arrow shows that the activity has been decreased. Now we are going to see the reasons why this increase in temperature increases the activity, or it actually denatures or decreases the activity after optimum temperature. The reasons are very important. When we increase the temperature, what happens? Reactants are made up of some of the molecules or some of the atoms. When we increase the temperature, the kinetic energy of those atoms increases, and those atoms are converted into products. Activity, kinetic energy increases, their vibration increases, and this converts the reactants into products, right? At optimum temperature, their activity is maximum or their vibrations or collisions are maximum. When we increase the temperature beyond this optimum temperature, the vibrations of the atoms of the enzyme molecules vibrations also increases, due to which the shape or the three-dimensional structure of the enzyme is destroyed, or its active site is destroyed, into which substrate can no longer fit and no reaction can occur. This is the reason that the reaction rate decreases after optimum temperature. Now, let me give you one example. When we boil an egg or fry an egg, what happens? Does its shape changes? When we boil an egg, it means we are providing it high temperature. And when we fry an egg, again, frying pan also has a very high temperature. So when we pour egg onto the frying pan, egg is basically protein in nature, right? Egg white is protein in nature, and its yolk has carbohydrates, proteins, and fats. When we pour this egg onto the frying pan, what happens? That is permanently changed. Its shape is permanently changed, right? So same happens with the enzymes, because enzymes are also protein in nature. So when the temperature increases beyond optimum temperature, what happens? Its three-dimensional globular structure or its shape is permanently changed, into which the substrate can no longer fit. Its active site is also lost, and the substrate can no longer fit, and the reaction of products cannot be formed. Reaction cannot occur and products cannot be formed. So this point is known as the denaturation of the enzyme. When the three-dimensional globular structure is lost after optimum temperature or at high temperature, enzymes are said to be denatured. Enzymes are actually destroyed, and for enzymes, we do not use the word destroyed. There is a term denaturation which is used for the enzymes. Understood?
Excuse me, miss.
Yes.
It's like if the egg contains protein and enzymes also contain proteins. So
Yes.
when the heat temperature is high, when the temperature is high, the protein state changes or the food that is going to be react with the enzyme.
Protein, actually, enzyme shape is changed. It is denatured, and this active site which we have studied is no longer available. It might change into any other shape in which the substrate will not be able to fit or fix.
Okay.
Okay, so that's why enzyme is said to be denatured, and our reaction rate also goes down. The reason is because enzymes are not available for the substrate to attach. That's why products will not be formed. Okay, let me
Okay.
revise again in just a few seconds. We studied at optimum temperature. It is a temperature at which the activity of enzyme will be maximum. It means that we will get maximum products. And every enzyme has its own optimum temperature. All enzymes do not work at one optimum temperature, and the enzymes which are working in human body, their optimum temperature is 37 degrees centigrade. And how the temperature increases the rate of reaction? When we increase the temperature, the collision or kinetic energy of the reaction increases, and the collisions of the substrate molecules increases, and they are converted into products. When we further increase the temperature beyond the optimum temperature, kinetic energy becomes so high, and the molecules of the enzymes also start colliding, and their vibrations also become so high that they are also converted, or they are denatured. Their three-dimensional globular structure is lost, and the active site into which substrate binds, that is also lost. So enzyme is said to be denatured, and reaction goes down, or the reaction rate slows down. Okay, beta. After optimum temperature, you have to remember that enzyme is denatured. Just one thing. Optimum temperature, enzymes are denatured. Denatured means that they are completely or permanently destroyed.
Miss, I'm not able to listen. Can you speak a little louder?
Okay. After optimum temperature, enzymes are denatured. Denatured means that they are permanently destroyed. And if we are going to slow down the temperature again, their activity will not be restored. They will not be able to carry out the reaction again because they are permanently denatured. You can take an example of egg again. If we have boiled the egg, its shape is permanently changed. And if we are going to pour that egg again into cold water, again it will not be converted into its original, you can say original shape or original consistency. Okay. So in the same way, if you slow down the temperature of the,
Miss,
yes,
when an enzyme is denatured, its state is changed. I mean,
yes,
when it is destroyed, what happens to the enzyme?
its physical state is also changed and chemically it is also destroyed because some of the bonds are broken down chemically, which results in its change in its shape.
Okay, Miss.
Okay, it's both physically and chemically it is changed. So the active site is lost and substrate can no longer fit, so reaction will not occur. You have to remember here one thing, that before or at low temperature, before optimum temperature or at below or at low temperature, enzyme is inactive. And after optimum temperature or at high temperature, enzyme is denatured. Denatured means destroyed. Its activity will not be restored. But inactive means that if you are going to increase the temperature, it will become active. Okay. We also place or preserve things into the freezer, refrigerator. So refrigerator has low temperature. If the bacteria are present into any food or fungi is present with the refrigerator, their enzymes are inactive in freezer. That's why they do not grow. And sometimes if the temperature becomes optimum, then they also starts growing. So in frozen food, you can say enzymes are inactive. They can be activated anytime when they are provided with the optimum temperature or the temperature increases. But once they are denatured at high temperature, then their activity will not be restored. Okay, in the same way, pH also affects the activity of enzyme. I think you all must be familiar with the pH. You might have done some of the experiments in class 7. There is pH ranging from 1 to 14, acidic and basic. You studied about acid base. Did you study?
No, Miss.
Okay.
Miss, we have studied about the temperature, but not about the pH.
Okay. Acid. Okay, you studied about acid or base. Any idea about the acid and base? Okay, acid is basically the negative log of hydrogen. Okay, that is the negative log of hydrogen. In other words, if you see that there is water present, right, and water can be split or broken down into its ions, it can be ionized into hydrogen ions. These are known as the hydrogen ions, and we also call them protons. And it's also broken down, it also splits into hydroxide ions. These are the hydroxide ions. OH negative is the hydroxide ion and H positive, it's known as the proton, and it's also known as the hydrogen ions. So pH is actually the negative log of this. Log you will study in maths, right? It's a negative log of hydrogen ions. This is actually the pH. Negative log, pH is equal to negative log of hydrogen ions. This is pH. Okay, in simple words, you can say if there are more protons, that substance molecule will be, or solution will be acidic. If it has more hydroxide ions, it will be basic or alkaline in nature. Have you heard about hydrochloric acid? That is acidic.
Yes.
Yes, Miss,
There is
that is present
sulfuric
in the
acid.
stomach.
Yeah, there is sulfuric acid, nitric acid, hydrochloric acid. So these acids have different properties. They are sour in nature and they are corrosive. They can corrode materials, and we also use acids at our home for washing floor, bath, for washing bathrooms, and for washing floors. So they are acidic in nature. In the other way, there are alkaline substances like sodium hydroxide is alkaline. Okay, this will be beyond the topic or
Excuse
complicated.
me, Miss.
Yes,
Miss, pH means temperature.
no, pH is not temperature. That is that tells you whether the substance is acidic or basic.
Okay,
Okay.
it means if pH is high, that the substance is acidic. If pH is
No, no,
low,
no,
that
no, no, no,
substance is
no,
not
no, no.
acidic.
It's not high, not low. There is a pH meter and pH scale which ranges from 1 to 14. Okay. It has values 1 to 14. 7 is neutral. 7 means that that substance which will have 7 pH is not acidic, it's nor basic, like water. Water has 7 pH. Water is not acidic, not basic, right? In the same way, if the substance has pH between 1 to 6, right, that will be acidic. So 1 to 6 is not high pH. It is basically low pH. 1 to 6 is low. 8 to 14 is high. Okay. If the substance has pH between 8 to 14, that is basic. Did you get this? There is a pH meter, there is a pH scale, pH paper, which is used to check whether the substance is acidic or basic. If its value is between 1 to 6, that substance is acidic. And if its value is between 8 to 14, that substance will be basic. Okay, you will study about this in chemistry in more detail. I think for now this is enough, sufficient which is needed. Acidic is 1 to 6, 7 is neutral, and basic substances will have pH from 8 to 14. And this is checked through the pH meter, and this acidic has red color and this has blue color. The more you go towards the 14, the more bluer color you will get, and the more you will move towards 1, 2, you will get more or brighter red color.
Excuse me, Miss.
Like your rainbow colors. When you go towards the 6, the red color will have the low
Excuse me, Miss.
intensity. Yes.
Miss, the 1 to 6 are more acidic than 8 to 14.
No, 14 will not be acidic, it will be basic. The substance which will have 1 pH will be more acidic than the substance which will have 6 pH.
Okay.
1 to 6 are acidic, means we are moving from 1 towards 6. 1 will be more acidic, 2 will be less, 3 will be lesser than 1, 4 will be lesser than 1, 5 will be more lesser than 1, and 6 will be least acidic as compared to 1. And
Okay.
pH means when we are moving towards the 14, the substance is getting more alkaline or more basic. If we are moving towards the 8, the substance is getting less alkaline. 8 will be less alkaline than 14. Okay, this is the pH, and this pH also affects the enzyme. In the similar way, like we studied about temperature, enzymes also work at optimum pH, and that is known as that is different for every enzyme. Some will work at 7 pH, some will work at 1, 2, 4, 6. Every enzyme has its optimum pH or a specific pH at which its activity is maximum, similar to temperature, and that is the optimum pH for that particular enzyme. Those enzymes which are working in our mouth, you studied about salivary amylase, its pH is 7. 6.8 you will see in some books, or it's 7. And the enzymes which are working in our stomach, it has 2 pH. Means 2 is acidic or basic. I've told you about the pH meter.
Miss acidic.
Yes.
Acidic.
2 means those enzymes which are working at 2 pH, they are acidic, and 7 will be acidic, basic, or neutral.
Miss neutral.
This is neutral, right? So the enzymes which are working in our mouth or in the saliva, they are neutral, and those which are working in your stomach, they are acidic, and those which are working in your small intestine, they work at 9 pH. So 9 is basically alkaline or basic. Okay, in the same way, they work at optimum temperature, or their activity is maximum at a temperature which is the optimum temperature. If the activity is maximum at optimum pH, if the pH is changed, if this enzyme which is in the stomach, it is 2, and if you change its pH to 8, which is alkaline, 12 or 14, which is alkaline, so this enzyme will be denatured. In the similar way, like it was denatured by changing the temperature, so it will be denatured, and this enzyme will be denatured, and its three-dimensional globular structure will again be lost, and it is said to be denatured or destroyed, and the reaction will not be able to proceed if it is denatured. Okay, I think you have understood about the pH 2. It is similar to enzyme. It is similar to temperature. Here you can see how we are going to find out the pH of the enzyme. Always remember that you will see a bell-shaped curve for temperature and bell-shaped curve for enzymes. Here you can see that this is this central line. The maximum peak is showing the optimum pH for that particular enzyme. Here is the pepsin. Pepsin is enzyme and its optimum pH is 2. For this green enzyme, its optimum pH comes to be almost 6.8. Okay, and for this blue enzyme, its optimum pH comes to be 8 or above 8. You can use the scale to find out the exact pH. Okay. Next is the effect of substrate. Substrate also affects the activity of enzyme. Okay, let me make it easier through some examples or figures. If you have substrate and enzyme present, then definitely reaction will occur or reaction will proceed. If one of them is missing, the reaction will not occur. Take an example. E is actually representing here enzyme and S is representing here substrate. If you have 100 units of enzymes, unit for enzyme is unit. Okay, if you have 100 units of enzymes and if you have 10 units of substrate, and if I say that one substrate will react with the one enzyme, so it means that 10 will react with 10 enzymes, we still have 90 enzymes left. If we are going to increase the concentration of the substrate to 20, then 30, 40, and so on, so our reaction will also increase because 30 will react with 30, 20 will react with 20, we will still have some enzymes left, and when the substrate will be 100, it means that all enzymes active site will be occupied by the 100 substrate. So our reaction will keep on increasing up till 100 units of substrate. Understood? Means we have kept the enzyme concentration constant, right? Enzyme concentration is constant and we are increasing the concentration of the substrate. Right? This reaction will increase till all the active sites of the substrate are occupied by the active site of the enzyme is occupied by the substrate. When there will be 100 units of substrate reacting with the 100 units of enzymes, then no active site is left to react with other substrate. If we are going to further increase the substrate by 110, will the reaction rate increase? Will the reaction rate increase? We have 100 enzymes which are already occupied by the 100 substrates, and we have increased the substrate by 10 units more. So will our reaction rate increase? Girls, I'm asking from you. Girls, Peter, can you hear me?
Yes, miss.
So I have asked, if we are going to further increase the substrate, will our reaction rate increase?
No, miss, it will decrease.
No, it will not decrease, but it will not increase. Okay, let me tell you this again. But we have 100 enzymes, and enzymes are kept constant, right? They are 100. We are not going to change its concentration. Actually, we are going to see the effect of substrate. So we will keep the enzymes constant. We will increase substrate. We can increase substrate by any figure you want. Like if they are 10, and if you have increased it by 10 folds or 2 folds, means you have made them 20, the reaction will increase because 20 will react with 20 and we will still have some of the enzymes left. So when we will increase its concentration to 30, our reaction rate will again increase because we still have 70 or 80 enzymes left. In the same way, it will keep on increasing up till 100 units. When there will be 100 substrate and there will be 100 enzymes, 100 substrate will react with the 100 enzymes, right? So it means that all active sites of the enzymes are occupied by the substrate. No free enzyme is available to react with the substrate. So if we are going to further increase the substrate, like we have increased the substrate from 100 to 110, right? So reaction rate will not increase because 100 are occupied by the 100 active sites, substrates are occupied by the 100 active sites, and those 10 which we have further increased, these 10 do not have active sites to react with. So reaction will not increase further. But it will also not go down. It will be constant. So in this case, we are going to get a graph like this. Activity is again on the y-axis and substrate concentration is on the x-axis. You can see here similar to this. Reaction is on this, and in this way, what happens when we increase substrate concentration, reaction goes upward, 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 and here. It means that 100 enzymes and 100 substrates both, right, they've reached their maximum, and when we further increase the substrate by 110 folds, 120 folds, the reaction goes straight. It will be straight line, it will not bend down. Like pH and temperature, it will go straight. It means that it is constant now. Constant means the reaction rate is not decreasing nor increasing. It is constant because all active sites are occupied by the substrate. Understood? This is the effect of substrate, and in the same way, enzyme also affects the activity of reaction or the rate of the reaction. When we will have to see the activity of enzyme, we will keep the concentration of the substrate constant. ठीक है, इसको हम बेटा रिवर्स इस तरह कर लेंगे. Then we will keep the concentration of the substrate constant. Substrate will be constant. Then let's take example here. When we will find out the activity or effect of enzyme on substrate, then we will take enzyme and substrate. Substrate will be constant now. Now we will increase the enzyme. And when we will increase the enzyme by 2 folds, 20, 40, 80, and 100. Up till 100, our reaction will increase. Yes.
मिस बाय अगर सबस्ट्रेट बढ़ाएंगे और एंजाइम नहीं बढ़ाएंगे तो रिएक्शन हमें लो मिलना चाहिए, हाई कैसे मिलेगा?
Sorry, I didn't understand. Come again.
मिस हम जब सबस्ट्रेट बढ़ा रहे हैं लेकिन हम एंजाइम नहीं बढ़ा रहे हैं तो हमें रिएक्शन लो मिलना चाहिए,